Now let's talk about the bioreactor monitoring systems and designs. So let's look at different methods that can be used. So one of the method to monitor these bioreactors is called as the stirred bank bioreactor. It is the most common form of fermenter. This is used commercially. And if you look at the presence of different supporting things within this fermenter, they include an agitator, baffles, includes aeration spargers for aerobic culture. It would be producing gases within the substrates to facilitate the growth of microorganisms. Then the probes, sterilizable monitoring probes. The probes that can give you information about the substrate, and they should be able to be sterilized, because if they are not sterilized, they can be a source of contamination. Similarly, dissolved oxygen, temperature, and foam, anti-foam, filling systems, and draining ports. These are all things that make the component parts of a fermenter, especially the stirred tank bioreactor type of fermenter. As I said, this is very much commonly used. So if you look at why this agitation is required in this reactor, there are many reasons. If there is a constant agitation and consistent one, it would help in temperature control, pH adjustment, also oxygen absorption onto liquid medium, and overall culture health. We can mix all the ingredients together and nutrients together, so that would be actually helpful. So this reactor agitation has got a very important role to play for the normal growth and development of these fermenting microorganisms. We need to look at constantly, we need to be able to look at this fermentation health in real time, and we should have proper monitoring system to know that whether this fermentation process is going smoothly, the health of microorganisms is good enough, and the production of different desirable metabolites is taking place, and their productivity is good enough. So these are the things that we have to monitor in all these bioreactors, and that can tell us about the efficiency of our process, bioreactor process or fermentation process. So there are many methods in which we can measure or monitor the microbial growth patterns within this fermenter. I think the most common ones are the indirect methods of measurement of biomass by absorbance at 620 nanometer in a spectrophotometer. So this is a good method or good way to measure or quantify these microorganisms that are present, and they are viable within these fermentation tanks. So this can be utilized as a normal method, as we would be making standard curve first of all of these microbes, and look at the absorbance value at different concentration on this standard curve. How we make this standard curve? We have to look at simple. We have to start with the fresh microorganism, because we have to have this as a standard curve. So through centrifugation, we would be taking these microorganisms from the log phase. We would wash these cells with 0.1 molar ammonium sulfate at pH 7 buffers, and then we serially dilute these pellets. So serially diluting these pellets would give us different concentration of these microorganisms, and once we can measure the absorbance of at different concentration of these microorganisms, we can develop our own standard curve. Then we can look at this actual dry weight biomass that is measured in gram per liter. It would be determined through dilution by direct biomass measurement after oven drying of each dilution in pre-weighed boards at 70 degrees C for 24 to 36 hours. So this is actually the conditions in which we would measure this dry weight biomass of these compounds, these microorganisms. We have to, of course, as like any other experiment, repeat this in triplicate. We would be washing this biomass with water to avoid this influence of culture medium, because that might have some turbidity in it, and that is going to interfere with the absorption when we measure this value through spectrophotometer. So that effect can be minimized by washing with water to remove this culture medium. So once we have this standard curve, by plotting absorbance versus the dry mass weight, then we can quickly and reliably measure this total biomass in our actual biofermenter. And the value is given in gram per liter. Now the next thing, if you have to look at the health of these microorganisms, viable cell count can be measured by flow cytometers. So through flow cytometry, we can measure how many cells are viable within that culture tank within this bioreactor. We normally produce this two-color histogram. This is collected by gating these bacteria, two bacteria only population, and two parameters are seen by scattering this light scattering distribution, and we get two colors. One is green, that gives us the live bacteria. Another is red, that gives us dead bacteria, and then there is a combination of these two colors that gives us information that these are the stressed bacteria. So in this we can actually quantify and look at the ratio of live organisms, dead organisms, or the microorganisms which are stressed in the real-time fermentation. Then we have to look at some other criteria such as substrate consumption and product formation rates. We would be more interested in the end product. So we would like to know that how much is the product being manufactured, being produced, what is the rate of production of this product. And this is possible through HPLC. This is one of the method to actually measure different end products and their analogs as well. Or we can also use membrane-bound enzyme biosensors. They are also utilized to do this kind of analysis, and it takes 20 to 1 minutes to run respectively. So HPLC would take 20 minutes, and this membrane-bound enzyme biosensors would take 1 minute. Of course, HPLC is more complicated. It takes more time. In real time, it would be less effective because we need to have information quickly, whether it might take more time in case of HPLC to analyze these compounds. But the good thing is that you can look at the relative compounds, similar compounds that are produced at the same run as well. Some of the compounds that can be measured, generally measured, are glucose, ethanol, maltose, lactic acid, lysine, etc. When we have to produce these products by these bioreactors, they are the one that can be easily measured. And normally sample preparation is simple. We have to use this 0.45 micron filtration and dilute it and look at the absorbance value. If this is above the range, we can further dilute this solution and get the desired value, actual value of these compounds within this culture medium. If you look at organic acid production, if that's our objective to produce organic acid, then there could be simpler methods. For example, if you are producing organic acids, the production of organic acid is increasing the acidity or decreasing the pH. If we can titrate this with the sodium hydroxide, the amount of alkali that is absorbed, judged by endpoint determination through indicator compounds, it can be easily seen that whether this is going up or down. Another thing that we want to monitor in these bioreactors, it could be microbial respiration, and it would depend again whether we are talking about aerobic cultures or anaerobic cultures. So if you are talking about these aerobic cultures, maybe we can monitor the oxygen, dissolved oxygen level through probes, or we can also measure this carbon dioxide in the exit gases. So that information would tell us about what is the rate of growth or the respiration rate of these microorganisms. So that can give us good information about the growth mechanism or the growth rate of these organisms. So if we have to do rapid analysis, as I said, that it is an online ongoing process. You have to do the live monitoring of bioreactor, and you have to take action if things go out of control. So it is very important to have the real-time information. So there has been a lot of new developments and research that is going into develop technologies which are more real-time, which can give you quick information. One of the option is we can use Fourier transform mid infrared FT-MIR spectrometry that can be used for few things such as lactic acid and ethanol fermentation. But this is an ongoing process. We are developing more and more better and robust technologies to measure and monitor the growth of these bioreactors, and that would be very helpful for further development of this industry. The method can be this method FT-MIR. It can be used real time online. It can be used for process control purposes as well. So this is a little about what are different things that we have to monitor in bioreactors, and what are the design thing in bioreactors. And these informations are very vital for running up a real life bioreactor and a real life commercial product. So these are some of the information that would guide you that what we need to understand in order to have commercial production of a specific compound from selected strains.